Meiosis is the second type of cell division that occurs during M phase of the cell cycle. I really like this micrograph because it shows images of cells at various stages of meiosis. And importantly, at the end of meiosis, we're going to see resulting 4 cells rather than 2 cells like we saw in mitosis. And so you can actually start to see the formation of 4 cells in some of these images. Meiosis 1 produces 2 cells, and then meiosis 2 will then produce 4 cells from the original 2 cells. Remember during interphase that chromosomes duplicate. I want to point out now the presence of homologous chromosomes. Homologous chromosomes have come from parents of the individual who's about to go through meiosis. The members of a pair of homologous chromosomes, one paternal and one maternal, duplicate during interphase, specifically during S phase of the cell cycle, to produce sister chromatids, and these 2 sister chromatids make up 1 chromosome. So this cell has a diploid number of 2. Diploid meaning 2 copies of each chromosome, and then 2 is indicating that there are 2 total chromosomes. Meiosis 1 is going to separate homologous chromosomes from each other, and meiosis 2 will then separate sister chromatids from each other, so that the individual who's going through meiosis to produce gametes will give either the maternal or paternal chromosome that they received from their parents to their own offspring. So meiosis separates homologous chromosomes and sister chromatids to produce 4 haploid cells. Importantly, meiosis 1 is the separation of homologous chromosomes, and meiosis 2 is the separation of sister chromatids. The 4 daughter cells contain half of the genetic information that the original parent cell contained. A specific half, 1 copy of each pair of homologous chromosomes. Homologous chromosomes have the same order and type of genes along their length. These sister chromatids are produced by DNA replication or duplication of the 2 homologous chromosomes within the pair. One of these chromosomes is maternally inherited, one of these is paternally inherited. And the difference between these 2 is that they carry different alleles of the same genes. Alleles are alternative forms of genes. So for example, maybe along the length of this chromosome there's a gene for eye color. Both mom and dad give an individual the gene for eye color, but they may say slightly different things. The maternal chromosome may carry the allele for green eyes and the paternal chromosome the allele for brown eyes. Now, whether this individual has green or brown eyes depends on how the alleles interact with each other, and that's for a couple of modules from now. We're not going to talk about that now, but know that these homologous chromosomes, despite having the same order and type of genes, can have different alleles of those genes, or alternative forms of the genes. To go back to this image, I want to remind you of the distinction between diploid and haploid. When a cell has 2 members of a pair, the cell is diploid, and that's what most of our cells are, including those that are about to undergo meiosis. Meiosis separates the homologous chromosomes from each other, and so as soon as these 2 chromosomes have separated from each other, we immediately have haploid cells. 1 copy of each chromosome. Remember that 2 sister chromatids are not 2 chromosomes. The 2 sister chromatids connected are 1 chromosome, and so we immediately have haploid cells by the time meiosis 1 is done. Meiosis 2, with the separation of sister chromatids, just creates 2 haploid cells. 2 haploid cells for each original haploid cell produced by meiosis 1. This is the process that leads to the formation of gametes, haploid gametes that can be used in sexual reproduction. In females, we produce eggs with 1 copy of every chromosome. Males produce sperm with 1 copy of every chromosome. When these two join together through fertilization, we then have a resulting diploid cell with one copy of each chromosome. This diploid cell has one maternal and one paternal chromosome for each chromosome pair, and mitosis will result in the adult. The phases of meiosis are similar to the phases of mitosis. There's no pro-metaphase, but we do have the prophase, metaphase, anaphase sequence of phases like we saw in mitosis. Prophase involves the same condensation of chromosomes and disruption of the nuclear envelope. But what makes prophase unique, prophase 1, of meiosis 1, is that these chromosome pairs line up together. They form a tetrad, and they cross over, which means they exchange pieces of DNA. So in meiosis, we actually mix up our maternal and paternal DNA, and the crossing over is reciprocal and in the exact same place, so that we can give different combinations of genes to our offspring. Note the results of crossing over here. So what makes it unique is the pairing and also the crossing over. In metaphase 1, homologous chromosomes line up together on the metaphase plate. That's different than in mitosis because they lined up single file on the plate in mitosis. Anaphase is the actual physical separation of homologous chromosomes from each other. It's called anaphase 1 because this is the anaphase in meiosis 1. And here we have the actual separation of homologous chromosomes. A telophase can result in the separation of two whole cells, but very often telophase doesn't occur. Instead, the cells immediately go into prophase 2. And prophase 2 has no crossing over and no pairing of homologous chromosomes because we have separated the homologous chromosomes from each other. In metaphase 2, chromosomes line up single file like they did in mitosis, but notice we've reduced the chromosome number by half. Anaphase 2 is the separation of sister chromatids, which is how we could describe mitosis as well, separation of sister chromatids. Telophase 2 and cytokinesis will result then in 4 haploid cells. The 4 haploid cells have one copy of every chromosome. The sexual reproduction allows organisms to recombine alleles of genes to make different, potentially better combinations. That's the benefit of sexual reproduction. The benefit of sexual reproduction is to take genes or alleles of genes from two different individuals and create new combinations so that the offspring may be better suited for the environment. This is something that asexual reproduction can't do because the offspring of asexual reproduction are genetic clones of their parent. And so there's no combination or potentially better arrangement of genes better fit for the environment. The sources of variation include crossing over, which we've discussed already, and also independent assortment. An independent assortment is the random alignment of homologous chromosomes in meiosis 1. Let's go through independent assortment first. Here we have a cell that is 2n equals 4, or has a diploid number of 4 total chromosomes. The gametes will be n equals 2, haploid number of 2 total chromosomes. In metaphase 1, if all the paternal chromosomes line up on one side and all the maternal chromosomes line up on the other, then the resulting gametes will either contain all paternal or all maternal chromosomes. Now, if another cell goes through meiosis, which another cell will, these chromosomes can line up randomly, and so here we have paternal chromosomes lining up on opposite sides of the plate. In that case now, the resulting haploid gametes still have one copy of every chromosome, but notice that it's a combination of paternal and maternal chromosomes. The number of possible combinations that an individual can make is based on the number of chromosome pairs. 2 to the n, where n is the number of chromosome pairs. Because there are 2 chromosome pairs here, The number of unique gamete types, unique combinations of chromosomes, is 4. One that's all paternal, one that's all maternal, one that has this combination of maternal and paternal chromosomes, and one that has a different combination. We have 23 pairs of chromosomes. So as humans, we can make 2 to the 23rd different unique gamete types with only one type containing all paternal and one type containing all maternal. So basically, if I'm going through meiosis, there are 8 million and some different combinations of maternal and paternal combinations I can make, but only 2 of those have just paternal or maternal. All the rest are different combinations of maternal and paternal chromosomes. Let's apply this to alleles. Remember that along the length of chromosomes there are hundreds of genes. And in this situation, the maternal chromosome that I received from my mom is carrying the brown eye allele. The paternal chromosome is carrying the green eye allele. On another pair of chromosomes, there is a gene for hair color. The maternal chromosome is carrying the black hair allele. The paternal chromosome is carrying the red hair allele. Independent assortment shows 2 different alignments of these 2 pairs of chromosomes. And of course, I'm ignoring the fact that I actually have 23 pairs of chromosomes. I'm just pretending that I have 2 pairs, so that we can look at these 2 genes and the independent assortment of these alleles of the genes. With this arrangement, I can give my offspring brown eyes and black hair, or green eyes and red hair. But independent assortment allows me to mix up what my parents have given me and mix up what then I give to my offspring. In this case, I give my offspring brown hair and red hair, brown eyes and red hair. In this case, green eyes and black hair. And of course, whatever the other parent contributes to our offspring will also matter as far as whether the offspring has green eyes, black hair, brown eyes, or red hair. Crossing over is the other source of variation. And crossing over involves taking pieces of homologous chromosomes and switching places. So on this pair of chromosomes, I have, among the thousands of genes along the length of the chromosome, a coat color gene in mice and an eye color gene in mice. One allele is the brown hair allele, or brown coat allele. The other is carrying a white coat allele. And then at another gene location, there is an allele for black eyes or an allele for pink eyes. Notice that mom gave a certain combination of alleles and dad gave another. Without crossing over, this mouse can either give brown coat and black eyes or white coat and pink eyes. But with crossing over, which involves the crossing over or recombination of DNA between sister chromatids, we can get a recombination of DNA. In the tetrad, a chiasma is formed, and a chiasma is where we actually get a cleavage of DNA. The breakage or cleavage of DNA is repaired, but because of the chiasma, the repair puts the 2 alleles of the 2 homologous chromosomes in different locations, so that the result is one chromosome carrying the brown, I can't remember the names of the alleles right now, the white and pink alleles, the other carrying the brown and black alleles. White and pink alleles and brown and black. Because of crossing over, we've recombined alleles, and this is completely separate from independent assortment. There isn't any good formula for predicting the different combinations based on crossing over, because these chiasma can form anywhere along the length of the chromosome. We're not disrupting any genes or putting them in different places. We're just switching what these chromosomes are carrying as far as the alleles of genes. So, by the end of crossing over, we have some parental type chromosomes which is indicated by the arrangement of the alleles in the original parent. These two are parental type chromosomes. And we also have what are called recombinant. Recombinant is indicating that we've mixed DNA from two different sources, in this case maternal and paternal DNA. What I want you guys to do is go back over mitosis and meiosis and fill in the blanks in comparing the two types of cell division. You'll come up with an answer for the number of duplications in mitosis and meiosis, number of cell divisions, number of daughter cells, and so on. Take a few minutes to fill out the table, and then we will look at the answers as soon as you're done. Just go ahead and push pause on the video. In mitosis and meiosis, there's only one round of DNA replication during S phase. In M phase, the number of cell divisions is 1 in mitosis, but 2 in meiosis, so that we produce 2 daughter cells in mitosis and 4 daughter cells in meiosis. The number of chromosomes in the daughter cells in mitosis is equal to the number of chromosomes originally in the parent cell. In meiosis, we reduce the chromosome number by half, so in all 4 cells we only have 23 total chromosomes. In mitosis, the chromosomes line up single file on the metaphase plate, but in meiosis 1, they line up as pairs. In meiosis 2, they line up single file. If I compare the daughter cells to the parent cell, remember that daughter cells in mitosis are genetically identical to the parent cell, while daughter cells in meiosis have half of the genetic information, 1 copy of every chromosome. And the reason for these two types of cell division, mitosis and meiosis, growth and cell replacement and for mitosis, and sexual reproduction for meiosis.